All right, well, tonight, our very own board member, Will Peterman, is going to be our speaker. And I'm just going to read what was in the introduction in the email. So you may have already seen this, but that Will spent 8 years monitoring bee populations at weedy sites around central Puget Sound. I just heard someone call it Puget. That was bad. The central Puget Sound, most notably at an abandoned golf course at the end of the runway at SeaTac Airport. And so he's going to be sharing some anecdotes about nets, traps, and boxes full of bees. So I'm looking forward to hearing it. Will, it's all yours.
All right. For 46 years, runway 34R at the Seattle Tacoma International Airport ended in a golf course. In 2014, the Tyee Valley Golf Club lost their lease, and management of the area fell to the Port of Seattle, which immediately began a program of habitat restoration. Now, the primary goal of the project was water management. Runoff from the airport creates a couple of tributaries to Des Moines Creek, which runs across south of the airport and down to the Sound. And a secondary goal was wildlife management, and specifically the port wanted to create habitat that would support predatory birds whose flight habits would keep them below the flight path of air traffic. So basically, they wanted to convert the old water features into a healthy riparian habitat and convert the fairways into something like the old indigenous prairie. So enter Bob Redmond and the Common Acre. Bob was already doing some work with honeybees near the airport, and he convinced the port to include pollinator conservation as one of their restoration goals. Now, the goal was to track the changes in the bee populations relative to the landscape interventions that they were planning to do. If the restoration helped the bees, we all wanted to know. And I joined the project in its second year in 2015, initially as a photographer and then as a field worker. And I wound up getting involved in the program design, specifically applying the lessons that we learned from the other project stakeholders, the port and the beekeepers and the coyotes and the wind. And I also wound up doing a lot of the lab work. And along the way, I became reasonably good at identifying bees. Just to be clear, though, reasonably good means that I still have a lot to learn. So, the take home, it didn't quite work. For the next bit, I'm going to give a quick description of how the survey protocol worked and was intended to work. Then I'm going to go over some of the challenges that we ran into along the way. Then I'll tell a couple of stories and then try to wrap things up, and then I'll take questions. And if you have any questions along the way, please put them into the chat. I can't see the chat, but I'm going to ask Colleen to interrupt me from time to time.
Will do.
And if there's an awkward silence at the end, I will start throwing pretty pictures at the screen until we run out of time. So now you know my plan. Okay, the process was this. We mapped out a set of transects, which are literally just straight lines from one place to another place. And we would walk from one end to the other, and at 5 points along the way, we'd stop and put down traps. Now you're looking here at 3 bowl traps, yellow, white, and blue, and 1 blue vein trap in the middle. And the blue vein traps catch a lot of bees. They all catch a slightly different set of bees, but the blue vein traps are more trappy than the bowls. So we didn't put down as many on each transect. We only put down 3. And each bowl trap got about 1 centimeter of soapy water in the bottom, and the vein traps got 2, maybe 3. And then we would supplement the trap collection with net collecting. And sweeping is important because it picks up bees that don't fly into traps, but it's also tremendously variable. At any given site, you're only around to sweep the net for at most 15 minutes before you have to move on. And you're completely at the mercy of whatever the weather is like right now. So if something is out flying earlier or later in the day, or only when the overcast breaks, you'll never know. And the most repeatable elements of the survey protocol are the trap transects. So we chose to emphasize the consistency in trap placement and trap collection over time spent with a net. And after 24 hours, we'd come back and pick them up. And we'd put the bees from each transect into a vial full of alcohol and take them back to the lab. And we would have liked to break up the collection in a more granular way. So, keeping the 5 stations on the transect separate, keeping the color bowls separate. But that level of detail adds a huge amount of effort to the field work. You're carrying just hundreds of vials, up and down the hills. And we weren't able to make the timing work. We did keep more detail on the net collections. So at a minimum, you need to record the plant that you're sweeping. And it turns out that once you're doing that, you might as well write down everything else. But again, the record keeping reduces the amount of time available for the actual sweeping. And then at the lab, the ones that were easy to ID stayed in alcohol for long-term storage. And the ones that we needed a closer look at, and there were a lot of those went onto insect pins with labels that show the transect location and the date they're collected and the person who collected them. And a unique number so we can tell the specimens apart over time. And then we tried to figure out what they were. And that was and is an interesting process, and we're still not finished. And I will have more to say about that later on. So that was the basic plan. As always, though, the devil's in the details. So what did we learn? And more to the point, what did we learn from? Now, the first set of problems came from other people. I'm not going to really get into it, but I will throw out a couple of examples. So the sticks in the corner here, underneath the big tree, are a test plot of willow saplings. The plan was that most of the labor for the restoration project would be done by the airport maintenance team. When there's nothing wrong with the runway, they have time on their hands, and it's kind of a treat to go out and work in the field like this. So they planted a lot of these test plots, and they were about to install the irrigation system to keep them alive, and something went wrong with the runway. And I'm sure you can see where I'm going with this. The saplings here are not alive. And then there was the time that we got some support from Boeing. And a few months, that was cool, but a few months after, they grounded the 737 MAX fleet. And we didn't hear much from Boeing after that. So most of the things that happened with our partners were in areas that we probably couldn't control. The best we could have done was to improve our contingency planning. And nothing really happened that a good project manager couldn't have guessed in advance. So, lesson learned from that, run the plan by a project manager. It helps. But the biggest problem is just that tracking bees is hard. It takes a lot of time, and it's not a full-time job. The people doing the work need to fit the field work into their existing schedules, and that's a real challenge. Now, we had funding to sample each transect 5 times a year, but there's interesting stuff going on from the beginning of March straight through to the end of September, at least. So 5 samples spread out across 7 months. So one example, one of the major pollen sources throughout the riparian areas, that is the bits that didn't need irrigation, is willow. And willow blooms really early in general, but definitely here. And we want to get the first transect out in time to catch the willow bloom, but we didn't always make it. And at the other end of the year, quite a few species of bee radically change their behavior when the food starts to give out. Bumblebees start churning out males. These little sweat bees start sending huge clouds of workers out to anything that looks like food. And if we want to capture the full story, we need to stick around for the epilogue. But in between, some bees just aren't active for very long. Their foraging season may only last for a week or two. So if you let 6 weeks go by between transects, those bees are going to come and go just completely undetected. And Andrena, the mining bees, those are particularly prone to this. We have at least a half a dozen species that we only detected on a single day out of 8 years. Now, sometimes there were a lot of them, but it was just on that one day, and we never saw them again. And there's a pretty good chance that there are several more species that are in residence there that we just missed. So did we learn any lessons from that? And well, a few. For one thing, from the beginning, we tried to space our survey dates evenly throughout the year, or throughout the season at any rate. And that was probably a mistake. It would have been better to front load the surveys so that we would have had more data from April and May and less data from August. And also, we should have had more money. I mean, just for the record. And the next big issue is the weather. This is a bowl that was frozen in the morning. And that didn't happen very often, but it did happen. And more generally, the weather drove our survey schedule all the time. Now, I'm not going to say a lot about that because it's kind of obvious. You know, a survey day in the rain is just a wasted trip. The bees stay home. I mean, it might be worth sampling anyway if there were rain specialists out there, so that finding the specialists might balance out the overall loss of results. But as far as I can tell, there aren't any. So we tried a couple of ways to adapt to the weather. And what we finally wound up doing was starting the survey as early as we could, and then setting a minimum interval, about 28 days, between surveys. And after 28 days, we would let the schedule slip to the next available window of reasonable weather. And by the end of the season, the accumulated delays would push the last survey out into the transitional period when the floral resources were starting to fade. And it worked. But again, if anyone asks, I might tweak the minimum interval so that they're shorter earlier in the season and longer late. Another problem is grass. Now, this one may be surprising, but when you're sampling a transect multiple times over months and years, it's important to do it in more or less the same way every time. And one of the challenges there is that from month to month and year to year, the ground can literally disappear. You know, if this bowl were at ground level, no bee would ever see it. And we might catch some spiders, but that would be about it. And it can also flood. And bowl traps don't do very well in standing water. So we spent a lot of time trying to build better mouse traps or bee traps or platforms for quickly setting up bowl traps in unfriendly terrain. And if you remember the earlier picture that showed the bowls and the blue vein trap out on the ground, you may have noticed that the traps there were spread out about 1 meter apart. And that's actually better. You know, we ran some field tests, and when you place the bowls together like this, it actually reduces the catch by about 10%. But it's a lot more practical to put all 3 bowls on a single post in bad terrain, and that high visibility bar in the middle made the grounds crew happy. They were really, really nervous that they might be out mowing or something and run over a piece of rebar. It's a bad day for everybody. And then there's the risk of overkill. Now, some of you may recognize this. It's Bombus occidentalis, the western bumblebee. And specifically, it's the last one that I ever saw in the Puget Sound lowlands. It was on the 22nd day of July in 2015, and it was less than a mile from one of our survey sites. Now, in general, field tests suggest that sampling at the level that we're using doesn't affect the long-term health of insect populations if their health isn't already critical, but this one is, or was. And the issue's not settled, but we did change some of our protocols on sites that were near recent populations of western bumblebees. Mostly we used fewer blue vein traps, since those tend to pull in bumblebee queens. And as it turned out, we never collected one. We never saw one in flight. So either the change worked or they were already gone. Then there's the wildlife. Now, apologies for the picture quality here, but there's a little drama. And you might be able to make out some of the players. On the left is one of our bowl traps, a little blue bowl trap. And on the right is a killdeer. And killdeer are insectivores. Now, I think that's a guilty face. The bowl was empty when I got there, and I'm not sure it was when he did. And we also had crows, steel traps, just to go play with them. And on one site, we had to start putting the traps up on posts just to keep the coyotes from taking a crap in them. It turns out that coyotes can aim. I'll spare you the picture of that one. Now, I'm not sure that we really learned anything specific from all that, except to just respect the locals. The details always depend on the local wildlife, and they will always surprise you. And here's a problem that may surprise you as well, and that's the traps themselves. So bowl traps are more or less standard in the bee research community, but there's no standard manufacturer. They're little plastic cups, usually Solo brand, that are treated with whatever paints are available. And we try to be consistent, but sometimes things go on and off the market. And it happened to us. In the middle of the survey, Solo changed their plastic cups. And this is kind of subtle, but I want you to pay attention to the blue and white bowls on the bottom right. What we've done here with this image, this is a photograph taken with a multispectral camera through a set of filters that correspond to the color receptors in a human eye. So we're photographing the bowls the way that they appear to us. And then we swapped out the filters for filters that correspond to bees. So this is what they look like to a bee. And you'll notice that all of a sudden, those two bottom bowls look a lot more alike. And it turns out that the new bowls don't reflect ultraviolet light. The only difference between the white bowls and the blue bowls is that the white bowls reflect red light and the blue ones don't. That makes them white to us. But bees can't see red light. So to a bee, they're pretty much the same. And anecdotally, we did notice a change when the bowls changed. We started catching more bees. And we think that the white cup was probably flawed from the start, because it was always the least attractive of all of the bowls. And bees don't seem to like a uniform UV reflector very much. Real flowers that reflect UV almost always have a dark spot in the middle, and the bees know that. And the bowls don't have them. And again, that's not really something that we can control. We use the standard trap so that we can compare our data to other people. But it's another source of, let's call it variability in the survey data. And finally, the last gorilla in the room, taxonomy. Putting names to the bees on the pins in the box. Now, naming bees is just hard. If any of you are on a naturalist, you've seen the way that bee people will argue with each other over a photo. And I'm here to tell you that giving them microscopes does not solve the problem. And in the last year, I've heard a lot of people talking about the taxonomic bottleneck in bee research, and it's totally real. And I don't have a lot to add to that conversation, except to note that some parts of the country are better off than others, and the Pacific Northwest is not great. For many of the groups, there simply are no readily available resources for telling them apart. The papers that do exist are old and copyrighted and focused on another part of the country and hard to find. So have a little sympathy for your local taxonomist. It's a tough job. And after 8 years, about 99% of our specimens have been identified to species, but that last percent is a doozy. And it's a very important percent for analyzing the changes in diversity over time. And our goal is to identify everything identifiable by into spring, and we'll see how it goes. Okay, those were the pitfalls. And if there's anyone in the audience who's interested in bee surveys, and I think there are, those are the kinds of things to watch out for, and I hope it helped. Now I'm going to tell couple, three stories. As it happens, they're all about sweat bees, although they're otherwise pretty different. And the first story is about Halictus tripartitus. It's the single most abundant bee in the survey area, and it's one of the smallest. And the first thing to know about tripartitus is that there are a lot of them. Something like 1 out of every 5 bees that we ever saw at the Port of Seattle was Halictus tripartitus. And the second thing to know is that they're almost certainly social. Their nest entrances are very busy places, and when you see one on a flower, you often see more. And the third thing to know is that, at least at the Port of Seattle, and generally around Seattle, they're female. All of them. I have seen thousands of Halictus tripartitus. I have never seen a male. And it's worth pointing out that a skewed sex ratio is not really weird in itself, especially among the halictids. Other sweat bees regularly run 10 to 1, 20 to 1, all the time. But 5,000 to 1? 5,000 to 0? That's weird. And that's why it's so important to study the early and the late season effects, because there's really 4 possible explanations for the missing males. Either they fly too early for us to catch them, they fly too late for us to catch them, or they don't eat, or they don't exist. Now, tripartitus are active the entire season. We have never not found them at the old golf course. But in March, on our earliest transects, they are just getting started, with numbers only a tiny fraction of what they'll become. Now, if the males overwinter, if they emerge in the spring and then look for mates, that's when you'd expect to see them, and we don't. Now, another possibility is they fly late, but tripartitus have a really distinctive late season pattern. At the end of the season, when the food runs out, they leave their nests by the thousand in a desperate search for whatever food is left. And if the males fly late, that puts a hard limit on when they can leave. If they fly any later than that, they'll starve. But we don't see them then. We don't see them then either. Now, it's possible that the males just don't eat outside the nest. We know that tripartitus are social, but this would be honeybee level workers feeding the drones stuff. We can't rule it out, but as far as I can tell, nobody's ever seen anything like it, and it's not the kind of thing that sweat bees generally do. And the last possibility, and the one that I kind of suspect, is that there simply are no males. It is possible for bees to reproduce without males by parthenogenesis. As far as I know, it's never been seen in social bees, but it has been seen in sweat bees. So, at any rate, there's something cool going on with these tiny little sweat bees in the Seattle area. And without a full season survey, we'd never have known. But speaking of random cool things, this is a tripartitus on Scotch broom. Now, for such a showy plant, Scotch broom is remarkably useless as a pollinator plant. I've only ever seen 3 species visiting their flowers, and 2 of them are bumblebees. And this is the third. Now, the second story is about Halictus rubicundus. Now, she's big for a sweat bee, probably 2 or 3 times the length of tripartitus. And the story begins with a glitch in the matrix, or at least a glitch in the restoration schedule. The port had the bright idea to prepare a new area of pollinator habitat. And they were going to send in bulldozers, scrape off the top layer of soil, truck in some fresh soil, and spread wildflower seeds. Instant habitat. And they got through step 1. And then a runway broke, and they never came back. And by the time we found out about it, it was about 3 weeks later, the bare soil had started to grow, and it looked kind of like this. Or more precisely, it looked like this. Every one of those brown patches in this image is a nest. Every one is Halictus rubicundus, and those nests are not much more than a foot apart. The scraped area was about half the size of a football field. There were thousands of these things. So here's a little sweat bee protecting the entrance to her nest. And here who she's protecting it from. This is a blood bee, Sphacodes, visiting another nest, one with a less attentive mistress, and that's bad news for any eggs that are in there. And that was the really cool part of the story. In less than a month, you know, not only had thousands of sweat bees moved into this new habitat, it was a fully developed nesting ecology, complete with parasites, a full suite of parasites. This is a hoverfly, literally shooting eggs at the entrance of the nest below her. And one interesting point here is that as far as I can tell, all of our rubicundus are completely solitary. That's not true nationwide, but it seems to be true here. In all my observations, I only saw them interacting at a nest entrance once, and this is a photo of it. And I'm pretty sure that the bee on the right is lost. The one down in the hole never moved from her defensive spot, did not let the other one anywhere near the nest, and just sent her on her way. And the last story is about a green sweat bee. It's probably Agapostum texanus. And as you might notice, it's not a very good picture, but it's the best one I have from the golf course. And that's interesting because Agapostum texanus is one of the most common bees in Seattle. They're everywhere. They fly all season, and they were certainly there during the survey. We caught lots of them. The reason that I don't have a better picture is not because they weren't there. It's because I didn't take very many pictures. And that, oddly enough, illustrates the problem with net collection on a project like this. The problem with a net is the same as the problem with a personal camera. It isn't running all the time. You're only getting a little slice of the environment as you pass through it. And the less time you have to devote to it, the narrower that slice is. Sometimes you get lucky, and sometimes you get this. Okay, so told you our plan, talked about some of the pitfalls, told a couple of stories about my favorite sweat bees. So how did it go? What happened in the end? When we finish our story, what do I think that we will have learned? Well, if you ask the port, they will tell you that the restoration was a success. The wetlands are reasonably healthy. The riparian fauna are returning and doing well, and many of the willows survived. The water, flows off the runway when it needs to be gone. It flows down to the sound without breaking anything on the way. And as far as I know, we haven't lost any airliners to bird strikes, so they're happy. If you ask us, we'll tell you that the survey went well, all things considered. We've established a fairly good baseline for the diversity at a disturbed site in the Puget Sound lowlands. We found 25 genera, 108 named species so far, and we've recognized 63 morphogroups that we have not yet named. Now, many of those morphogroups will turn out to be variants of species that we've already named, but most likely 30 of them won't. So we expect the final species count to be about 140, including a few species that are new to the area, new to Washington, and one that may be new to science. We're still following up on that one. And the collection that we've assembled will be useful to Washington researchers, I hope, for years to come. It's much easier to put names to your own bees if you have something to compare it to. So that's all good. Now, what's missing are the elements that were meant to bring all those together. We have the bees, but we don't really have anything to compare them to. The restoration work turned out to be largely improvised. The controls were uncontrolled. So we can say, just as a matter of observation, that there were more bees at the end of the restoration than there were in the beginning. But we cannot say yet that it wasn't just luck. The difference is right at the edge of statistical significance, and I don't know right now which way it's going to tip. So, taking the good with the bad, was it worth it? I think it was, or I think it will be. I'm curious to see how people will use the collection, and I hope that the next round of optimists will learn from our mistakes. hell, I might be one of them. Oh, and the good and the bad, that's a joke here, y'all. Bumblebee queen and a kleptoparasite. I figured I'd say that out loud for the people in the back. And that's a lot. It's pretty pictures from here on out. I mean, some of them are cool pictures, but they don't go anywhere in particular. So
Well,
this is probably a good time to stop for questions.
well, this is great, Will. I really enjoyed your stories about the different bees and just the appreciated that you discussed the downfalls and problems of different survey methods and things that could, because I've run into that myself.